Thank you. Hi, I'm Clemens at the Max Planck Institute for Molecular Genetics. I'm going to tell a little bit about CAdir, a method for clustering and visualization of single-cell transcriptomics data. So if you have ever done any single-cell RNA-seq analysis, you probably have done a clustering and embedded it in a UMAP, just like that. But then the question very often is, like, is it actually a good clustering? Or would, for example, alternatively, this clustering with more fine clusters be better? Particularly if we compare it with the ground truth annotation we have in this case, we can see that neither of these clusterings really represents what's going on in the data. For example, you have this blue cluster kind of like split over to two hemispheres. So really, it's somewhat difficult to determine cluster quality purely by looking at UMAPs. To make matters worse, very often then you probably have performed a differential gene expression analysis on the clusters. And what you might have noticed is that some of the p-values are much, much lower than you would expect. Part of the reason for this is that actually clustering forces a separation in the data. And then if we reuse the same data to perform differential gene expression analysis and find the marker genes, we get an enrichment in statistically significant p-values. So for example, on the right, I clustered purely random data into two clusters. And if you then perform statistical tests, you will see that in the p-value histogram, actually there's this enrichment in p-values lower than 0.05, although we would expect actually none. So in order to tackle these two problems, we developed CAdir, which clusters both the cells and the genes simultaneously by their direction in correspondence analysis space. It also lets you evaluate your clusters based on so-called association plots, which I'm going to introduce in a second. And then you can use the co-clustered marker genes for downstream analysis, such as, for example, cell type annotation. If you're not familiar with correspondence analysis, it's a dimensionality reduction method, somewhat similar to PCA. And one of its hallmarks really is that you can embed both the cells and the genes into the same space. So you have, for example, the cells in this like large dots and the genes in the small gray ones. And what it's doing is it arranges the points along directions in space. So, and the further along a direction a cell or a gene is, the more highly associated it is with that directional cluster. This, of course, also holds up in higher dimensions. I unfortunately cannot show you a 50-dimensional space, but in 3 dimensions, we can, for example, perfectly represent these 4 clusters here. And if we're now interested in a specific cluster, let's say the lime green cells here on the right, can we describe this cluster, for example, by the direction they lie along? So if we simply put the direction now through this cluster, could we somehow visualize how well this direction actually describes our cells? And for this, we can use these association plots, which have been originally introduced by Ella Grolinska. And you can imagine like you have a plane in the higher dimensional space. And if you have a point on this plane, then the x-coordinate in the association plot is simply the length of the projection of the point onto the direction, and the y-coordinate is the distance of the point to the direction. Of course, we don't do this just for one point, but for all points, which you can kind of visualize as if you were like rotating this plane around the direction and kind of recording where the points fall on it. And if you do that, you will end up with a 2D embedding like this, where you have the clustered cells on the far right and the marker genes kind of like trailing towards the cluster. And all the other cells are found in an almost 90-degree angle on the left side. And optimally, what we would like to see is that our clustered cells and co-clustered genes have a very high x-coordinate and very low y-coordinate. And this is also, you can see here, where on the left I have a so-called good cluster with a good separation of the co-clustered cells with a high coordinate, x-coordinate and low y-coordinate, whereas on the right, you have the clustered cells have a much higher dispersion along the y coordinate. Now, how can we use these directions actually for clustering? I'll just leave out here now the genes for a second, just to make it a bit more easier to visualize. But if you have the directions, you can simply assign all the cells to the closest direction based on the orthogonal distance. In practice, however, of course, we don't know these directions. So we just start out by initializing them semi-randomly. And then, as mentioned above, we can simply assign all the cells to the closest direction based on their orthogonal distance. We then can update the directions by such that the new direction minimizes the distance to all the points in a cluster simply by using singular value decomposition, which can compute this very efficiently. And we can then simply repeat this procedure until we eventually arrive at the stable configuration. And if we do this, we start out with from our data set, we might end up with like 8 clusters as shown here. But we had to pick the number of clusters in the beginning. And optimally, like we don't usually know the number of clusters in our data, so it would be great if we could somehow infer how many cell types or clusters there are in the data. And in fact, we can do this by splitting and merging our clusters. So where we look at the angle between the cluster directions, and if they have a very small angle to each other, for example, we can start merging them together because they're very similar. And similarly, we can take a cluster, subcluster it into 2 new clusters, and if these 2 new subclusters have a very small angle to each other, large angle to each other, then we can split them and keep the new clusters. And what is nice is that CAdir can infer the cutoff angle for this procedure from the data through a randomization scheme. So it's not actually necessary for the user to supply. And if you do this, you end up with this iterative cluster split merge procedure where you might start out with a certain number of clusters, but then the algorithm will adjust the numbers based on how well they fit to the data. And just to visualize for you how this then looks like in association plots. So for example, on the left, you have an association plot with a cluster where it's split into 2 new ones because the angle is very large. And similarly, on the right, you have 2 clusters with a very small angle to each other, so they're being merged together. And if you do this, if we now still have our original 8 clusters, then through the splitting and merging, the algorithm then arrives at a different number of clusters. So for example, here you can see several steps in this graph where it either merged them or split a cluster. And just to put the genes now back into the picture again, we can in fact cluster them very similarly to the cells. We simply assign them to the direction with based on the closest orthogonal distance. But because not all genes are in fact cell type specific or cluster specific, we first filter the genes based on the radius from the origin, which is usually includes, for example, housekeeping genes or other cell type unspecific genes. The remaining marker genes then can be used for any downstream application. Now, how does this actually look like on real data? So here, for example, I clustered some limb muscle data from the Tabula Murus consortium, which you can see has been clustered into 7 clusters as represented by the association plots for each cluster. And if we zoom in into one of these clusters, cluster 3, for example, you can see that the clustered cells in like lime green and the co-clustered genes in orange are well separated from the other cells and genes. And if we now add the top 5 most highly associated genes into the picture, you might already see some marker genes that might be familiar, like CD19 or CD22. But if we compare this now more systematically with, for example, cell type specific marker gene sets, we can simply assign to each cluster a cell type for which we find the highest enrichment in the gene set. And if we do this, for example, we can find that this is unsurprisingly a B-cell cluster, and we can similarly annotate the remaining clusters. And if we compare this to the original clustering from the Tableau-Morse consortium, we can also see it fits fairly well, even though the annotation doesn't always perfectly fit. And for example, cluster 1 has been left unannotated. Another nice thing is that we can see actually other similar clusters in these association plots. So for example, here for the B cell cluster, we can see that the T cell and the immune cell cluster, their directions have actually a much lower angle to the B cell cluster than the other cells. And the same is true basically for the vascular endothelial cells, where the endothelial cluster direction has a lower angle to the cluster. In the beginning, I told you one of our goals was to actually help you evaluate cluster quality. And here, for example, I have clustered PBMC data. And if you look at the association plots for the different cell types, you can see immediately that cluster 5, for example, does not look like the other clusters. And if we zoom in a bit, we can actually tell that it's completely dominated by 2 outlier cells. And that if we, for example, if we remove these 2 cells, then this cluster would disappear and the remaining cells would be clustered with the other cell types. So it really helps you understand if there's a bad clustering or outliers in your data. In terms of runtime, we compared it to 7 other clustering algorithms that are commonly used. And it's at least in our comparison, it's the fastest algorithm. You can cluster roughly 600,000 cells in 30 minutes on a single processor. But we, of course, also did a much more comprehensive benchmarking where we compared it to other cell clustering algorithms, but also other bi-clustering algorithms, both on experimental data and simulated data. I unfortunately don't have time to go into all the details here, but suffice to say that it actually outperforms most of the tested algorithms, including some of these deep learning-based algorithms we tested. And yeah, with that, I hope I could convince you that CAdir is a very intuitive way to cluster both the cells and the genes simultaneously based on their direction. And that we can use this split and merge approach to infer the number of clusters in our data independently. And that the association plots that we can make based on these cluster directions help you actually evaluate how well the clusters describe your data. It's also extremely fast, so you can use it even on cell atlas size data sets. And if you want to try it out, you can install it as an R package from GitHub. And if you want more information, there's also a bioRxiv preprint paper. And yeah, with that, I just want to thank everyone in the Vingron lab, of course, particularly my supervisor, Martin. And if you're interested, you can also have a look at the lab GitHub where you find all the tools. Thank you.
Great. We have time for questions.
Hi. I think I missed the first part of your talk, but have you compared to matrix factorization approaches? I think there's some of them which can, if you give it a cell by gene matrix, can actually do clustering for the cells and the genes. Like, for instance, something like NMF with some constraints where the first matrix represents the clustering of your cells and the second matrix gives you the loadings of the gene. So there's lots of flavors of NMF, and I think some of them are specifically targeting and giving you cluster for the cells and for the genes.
So we haven't directly compared to NMF, but I think...
Not basic NMF.
Sorry?
Not just basic NMF.
I think we had in our bi-clustering comparison some method that was like matrix factorization based. I don't quite remember which one it is. You have it on the bottom right here. Maybe one of them rings a bell. But like specifically like any NMF-based methods, I don't remember actually having tested against.
So you compare?
I don't think we do currently. Like I don't think we have tried that.
Other questions? Yeah.
Hi, thanks a lot. You said that the angle which you use to split and merge your clusters is somehow set automatically. Can you elaborate a little bit on this? How does this work?
Yes,
You get a p-value?
So basically, we have two different ways, depending on how much computational time you have. But either you can randomize your data, and then we can make like an association plot like you see here. And we look at the quantile below which the cells or genes fall, like 99% of the genes fall above this line you can see here. And then we say basically everything below is at least somewhat specific for direction, and we can score based on this then. And this is also then the angle we use for the cutoff and scoring the genes.
If I understood it right, you applied mostly to single cell data. I work with spatial data, so sometimes I have contamination from cell nearby. Have you tested your algorithm on this type of data? And my second question is, could you use your algorithm to determine if the clustering is bad or if it's just there's like low variability in the data?
So for the first question, whether we tried it on spatial data, I have not tried this, but I maybe should do that. And if the second was if we can identify whether the clustering is bad or if it's just low variability. Did I get this right? So one, yes, I think we can, yeah, because basically what you would see in the association plots is that if it's just not very specific for direction, right, compared to the other cells, they're just not very separated, or there you have these clouds. Maybe I can go a bit back. Like, for example, the cluster on the top left would be also a candidate where you can see that it's just not very well separated. And it's also the scale on the x-axis varies between the different plots, right? So if there were lower variability, I would expect it to the x-axis to be lower, but you would still expect some separation actually.
Other questions?
Hi, thank you for your talk. Let's see if I can formulate this properly. I was thinking in the case where very often like you're working with someone that generated the data and they're expecting that specific cell type or whatever, like classically I've seen sometimes with normal clustering algorithms, it doesn't appear because you don't get that resolution. And then a common thing would be subsetting the big group and then redo a clustering. Is something like that would be useful with this method, or if it's not picked in a first round, would subsetting not change the result?
So subsetting and reclustering would be, I think, like would work the same as for other kind of data sets. Yeah, you can also tune the resolution. Like you can set also the angle for the splitting and merging by hand if you want finer resolution. So that's also an option.
Thank you.
Anything else? I actually had one question. When you're choosing the directions, it seems like your clustering algorithm is based on choosing these directions, and you have an iterative approach for doing this with random initialization. I was wondering if there's a specific objective function that you're optimizing for the directions, and if you can pose this as like a global optimization problem.
I thought about it. I don't have one currently, to be honest, but yeah, it would be very interesting, honestly, like doing it this way and trying to figure it out.
And then I guess the other one would be in terms of initialization, there are all these heuristics, like for k-means, there's a k-means plus heuristic, which
is exactly what we're doing currently, yes, yeah, basically.
Perfect, great. All right, well, let's thank the speaker and all the speakers of the session.